A human has about 100 trillion cells in his body. All these cells must act in perfect synchronization for the human body to function properly. Each cell has a very well defined role in the body, and any small deviation can lead to serious problems for the body. And then you cannot help but wonder, how was this coordination, so fine and so well regulated, possible? Once again, we were lucky that from the multitude of possibilities, we achieved a proper combination for us.
Let's go to the clip where you show what is happening in the cell and how it's programmed with this code.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Now, Stephen, looking at that wonderful clip, I bet you that 85% of our audience didn't understand what they were looking at. But they did pick up a couple things. First of all of that was happening in one single cell. All right? All of those little machines that you were looking at are in one little cell. They're interconnected. Then they saw code. Your point is, if that code's not right, those machines aren't going to do their jobs, because that code, that information, makes them do what they do. And the question you're raising is, where did this complex code come from?